Assalamu alaikum. We will continue with the session. So we reach up to the category and the stated element of non-specific body defense mechanism. So from that we have the antimicrobial chemicals also. From the antimicrobial chemicals we have something which is called the complement. The complement, which is a group of plasma protein that will lead to lyase the microorganism, they will lead to lysis of microorganism and enhance the phagocytosis by optimization and the interferon, fight the inflammatory process. So they will increase the inflammatory process. The interferon also, which is also protein released by the virus infected cell. So when the cell is infected by virus, that infected cell, it will release the interferon, which protect the unaffected cell, the uninfected cell tissues from the virus to take over and to get in the new cell. So the cell which is infected by virus, it will secrete interferon. That interferon, it will protect the other cell from the attack of the virus. The fluidic acid and the pH, as we said, they are inhibit the bacterial growth. In the urine, we have, it is an acidic media that also will prevent the growth of microorganism in the urinary system. Also, when we pass urine, it will clean out the urethra and prevent the ascending infection to our body. The fever, as we said, it is trigger and decrease the possibility of the bacterial growth. So the antimicrobial protein, as we said, antimicrobial protein enhance the innate defense by attacking microorganisms directly and hinder the reproduction of microorganisms. So it will decrease the reproduction of the microorganism. The most important of them, which is the complement protein, as we said, and the interferon. The complement and the interferon. So the complement protein, it refers to a variety of protein in our body. We have more than 20 protein in our plasma. Those protein are circulating in our blood, but in an inactive form. So when there is no need for them, when there is no infection, they are inactive. So the complement is activated when there is plasma protein encounter with foreign cell. So when foreign cells or foreign antigen is there, then those plasma protein, they will be activated and they will start to do their action, such as in case if bacterial invasion is there, if fungal invasion, so or the mismatch red blood cells even, if we are giving a different type of cell from other body. So a reaction will be there and they will be active, which are known as the complement fixation. So it will lead to complement fixation. The function of complement protein, it will lead to lyse microbes, it will lead to lysis of them, destruction of them, by forming membrane attach the complex, which is MAC, we call it the membrane attack complex. So it will form the membrane attacks complex, MAX, over them. This will enhance and facilitate the process of phagocytosis. So it's like putting a tag on those cells for the phagocyte in order to engulf them. So enhance the facilitate the phagocytosis, which is known as the opsonization. So this process, it is known as opsonization. So opsonization, it is the those protein will form the membrane attacking complex over the cell which is infected or bacteria. Then those opsonization, it will lead to that it will be attracted by attract the macrophage. So activation of the inflammatory response will be there, and the clearance of the immune complex will come to that area. So lysis bacteria by forming the MAC, here the MAC or complement, they are fixed to the cells. Then the macrophage will come and engulf the and do phagocytosis of the attacked cells. So they tag the pathogen to enhance recognition of them by macrophage and destruction by phagocytosis, which is known as optimization. And enhance also clearance of the antigen antibody complex there. The interferon, as we said, it is protein secreting by virus-infected cells. Interferon bind to the membrane receptor, so it will bind to the membrane receptor surrounding the healthy cell surface. So it will bind to the healthy surface. This binding stimulates the synthesis of protein that interfere with the ability of virus to multiply in the healthy cells. So it will prevent the virus from multiplication inside the healthy cell. So again, the interferon, it is secreted by the infected cells by virus. When our cell is infected by virus, you know that the virus is living inside the cell or outside the cell? Inside the cell. So the cell, and it will multiply inside the cell. It will use its DNA and the RNA to multiply, to formulate the new virus. So those cells, they will start to produce interferon. That interferon, it will go to the other cells and prevent the virus from multiplication inside those cells. The fever, mostly when we are getting infection, one of the signs is fever. So the fever, it is abnormally high body temperature in the systemic, which is from the systemic response to invasion by microorganisms. So most of when we are getting infection with microorganism, fever will be there. So the hypothalamus, you know that it is the thermostat of our body that regulates the body temperature, can be resetted in a higher response to biogen. So it will set to a higher degree. So chemical secretion by white blood cell will expose to foreign cells. So some immune or benefit of the fever, that it will decrease the growth and reproduction of bacteria and virus, because mostly bacteria and virus, they need the normal body temperature in order to multiply, which is the 37 degrees centigrade. Also, lower the amount of iron available to the bacteria by inhibit release of iron and zinc from the liver and spleen, which will be used by the bacteria. Enhance the neutrophils to migrate. In case of fever, it will migrate from the blood vessels to the infected tissues to fight the microorganism. Increase production of also superoxides, which can kill the invading microorganism, also microbes. Promote T lymphocyte production, so it will enhance the production of T lymphocyte that will fight the specific bacteria or virus. Fever also increase the speed of repair process, because fever metabolism will be more, and also more blood will come to the area, as we said in the inflammation, and more nutrient will be coming to the area, more oxygen will be coming to the area. So those are the benefits of fever. In the adaptive defense mechanism, the adaptive body defense mechanism are the body-specific defense system, or the third level of defense. So here the immune response is, the immune system response to the trigger, the antigen, by the antigen, which is the bacteria or whatever is it. So also it's triggered against the specific antigen, the immune response. The initial immune response also, which prime the body to react more vigorously to the later exposure with the same antigen. So for the first time, it will identify the microorganism or the antigen as a foreign material. It will start to produce antibody for them. This will take a longer time because it is new for them. So it needs recognition, and it needs start to build up a new antibodies for them. But later, if we expose to the same antigen, then our immune system will have a memory that can recognize it immediately, and the amount of antibodies which will be formed in this case, it will be high. So immediately it will be recognized for the second exposure, and the amount of antibodies, it will be more higher. There are three aspects of adaptive defense mechanism. It is antigen specific. So it is specific to the same antigen. It will not act with other antigen. Also, it is systemic. The immune is not restricted to the initial infection site. The antibodies will be circulating all over the body. It will be circulating in the blood. So it is systemic. It is not localized. The inflammatory process, it is localized to the site of infection. The third thing, it has memory. The memory, it will lead to the recognition of the virus or the bacteria in the second time and attacking them by the attack the phagocyte. So it recognizes, then makes stronger attack to the previously encountered pathogen or previously known pathogen. So those the characteristic of the adaptive defense mechanism, that it is antigen specific. It will act for specific antigen. It will not act for other antigen. So the antibody which is for the tuberculosis, it will not act for syphilis. It is systemic. Those antibodies are circulating in the blood, so they are there present in our plasma, and they are ready to attack those antigen if they're coming again. It has memory, so it will remember that type of infection of that type of antigen, and it will attack them immediately when they are coming, and it will enhance the immune system to produce more antibody in those cases. So those are the three characteristics of them. It has two arms, or it acts in two ways. We have the humoral immunity and we have the cellular immunity. So it has two parts. The humoral immunity, which is the antibody-mediated immunity, we call it. Antibody-mediated immunity. The antibody which is formed. The cellular immunity, which is the cell-mediated immunity. So it has two types, cell-mediated immunity and the antibodies-mediated immunity. The type of immunity, as we said, antibodies-mediated immunity, provided by antibodies present in the body fluid. So antibodies will be formed and it will be present there in the body fluid. The cellular immunity or cell-mediated immunity, it is the lymphocyte themselves defend their body and targets virus infected cells, cancer cells, and foreign grafts also. Action is either direct lysis of those cells or indirect. Indirect by releasing chemicals that enhance the inflammatory responses and activate other immune cells to act against those cells. What do we mean by antigen?
Anybody come with
Any
foreign body.
anything which is recognized by the body as foreign body or foreign material. So any substance which is capable to existing in the immune system and provoke an immune response. So the immune system will respond to them and it will be identified as an antigen. So example of common antigens. The common antigen, we have foreign proteins, which is the strongest one. So even for bacteria and virus, sometimes in preparing of the vaccine, they are preparing some of the protein which is used by, which is present there in those cells. So the body will form antibodies against those proteins. So whenever this bacteria or virus are coming inside, so it will be attacked by the antibodies which form there. Nucleic acid, large carbohydrate. So nucleic acid from other cells, type of cells. Large carbohydrate also. Sometimes lipids also, it is considered as a foreign material. Bones and grains. Because of that, sometimes we will have allergy due to bones and the grains during that period. Now it is the period for the grains. Microorganism. So all of them, they are considered as antigen. So the antigen, for the antigen, the body will form antibody with them which will attach to them and help in destruction of them. So antigen, there is also self-antigen. Self-antigen, there are protein molecule in our cells are recognized by the immune system as a self-antigen. So the body will not react against them because they are from our own body. When they will act against them, if we have problem with our immune system, sometimes our immune system will start to produce antibodies against our own protein. In this case, it is called autoimmune disease. Autoimmune disease. When our immune system attacked our own cells or our own antigen. But normally, this will not happen. Every one of us is having different tissue types. Yes, we are a human creature, we are having many similarity, but we have different tissue types. Because of that, you hear in tissue transplantation or organ transplantation, it cannot be done, cannot be taken from anybody. Before taking organs for transplant, nowadays mostly they are transferring either kidney or a portion of the liver. So before doing that, they are doing many type of tests for tissue typing in order to have a minimal difference in the tissue type. Because if the tissue type is highly different, in this case, the immune system will react and it might be fatal. So instead of helping the person, we are killing him by those tissues. Because the immune system, it will start to fight against those grafts or tissues. So they do not trigger the immune responses, but they are antigenic to other people. So they are antigen to other people, trigger the immune responses in case of transplant or what we call it the graft rejection or organ rejection. Now you heard sometimes that Mr. So-and-so, he got kidney transplant and reaction happened, then the process failed. In this case, they have to remove that tissues, otherwise it might kill him. In the cell, also we have crucial cells of adoptive system. We have, the lymphocyte. Lymphocyte respond to specific antigen, B lymphocyte, and we have T lymphocyte. So we have two types of cells, the T lymphocyte and B lymphocyte. They are responding to antigen. Antigen presenting cells also, we have ABCS, which help the lymphocyte but do not respond to the specific antigen. So they are helping the lymphocyte in recognition of the antigen. So the lymphocyte, from the previous session of blood, you remember that the lymphocyte arise from the lymphocytoplast at the red bone marrow. The T cells, then the T cell develop the immunocomplement ability to respond to and to bind to a specific antigen in the thymus gland. What is the thymus gland? Where is it present? Yes, it is in the thorax. And it will be large and small age. After that, it will shrink. So the cell-mediated will produce the thymus or cell-mediated immunity, or thymosin. The B cells develop the immunocomplement in the bone marrow and provide the humoral immunity. So they are the T lymphocyte in the thymus gland, it will be developed, completely developed. The B lymphocyte in the bone marrow itself. So the red bone marrow, it has the immature or native lymphocytes. Then the thymus gland, the lymph node and spleen and another lymphoid tissues, they are helping in the production or activation of the lymphocyte. and the bone marrow, it is the B lymphocyte. So the T lymphocyte in the thymus gland and lymphatic gland, and the bone marrow producing the B lymphocyte. We have the antigen presenting cells. The antigen presenting cell engulf antigen and then present the fragment of them in their own service. So they will be on their own service like signal flag. So like doing labeling of them, where they can be recognized by the C cell. So then they can recognized by the T cells, and they will be attacked by the T cells. So the major type of cell behaving as the antigen representative cells, which is the dendritic cells, the macrophage, and the B lymphocyte. Dendritic cells, macrophage, and B lymphocyte. So they are tagging the cells. So antigen representing cells, they are tagging the cells, and then that tag, it will be recognized by the macrophage, and then it will be attacked. The dendritic cells are antigen presenting cells, also known as accessory cells. They are widely distributed throughout the lymphoid organs and connective tissues. They are present in the connective tissues and lymphoid organs. Their main function is to processing antigen material and presenting it to the T cells. So they are presenting it to the T cells. They act as link messenger. So they are acting as link messenger between the innate immune system and the adaptive immune system. So they are working in between the innate immune system and the adaptive immune system. The phagocyte found also in the lymphoid organs and connective tissue, they will remain there. They will not go out of them, waiting for the antigen to come to them. So they are remaining in their site, waiting for the antigen. When the antigen is coming in toward them, they will attack that antigen and engulf foreign particles and act as antigen presenters to T cells. T cell, then it will get activated. So they will activate the T cells. Activate T cell to release chemical substances, which in turn, that chemical substances which is released from the T cells cause the macrophage to become killer. So the macrophage, it will become killer. It means that it will become more phagocytic and hungry to eat those antigens. So it will kill them. So this is the immunity and how it happens. Did you understand? Is it understandable? Any question?